And hi, I'm Chirag Patel. I'm a postdoc at Stanford University, Division of Systems Medicine, and I'd like to thank ISCB for having me talk here as a proceedings paper. And also thanks to all of you for hanging in there. This is the last session of the last day. So maybe let's keep this interesting. I have a Twitter username here. Just tweet me up. Hopefully I'll keep you entertained for the last half an hour. So as you know, common disease like type 2 diabetes are a function of the stuff that we get from mom and dad, the heritable components, but often not talked about too much, especially in computational biology settings like this one today, are environmental factors. So things that the complex system of exposures that we encounter every day, like the foods we eat, the behaviors that we conduct, and even pollution measures that we're exposed to on a daily basis. So we have that complex system, and we have the heritable components that make up diseases like type 2 diabetes. And we're talking many, many different factors, many different genetic and environmental factors. All are hypothesized to play a role in diseases like type 2 diabetes. But yet we continuously investigate these in siloed environments. So as you know, to investigate the heritable components, we have this study known as the genome-wide association study to elucidate basically the genetic variants that are associated with diseases like type 2 diabetes. And recently, environmental epidemiology has also been looking at handfuls of factors like diet and pollution and so forth, also in association with diseases like type 2 diabetes. And actually, we have also put forth this new framework of studying environmental factors called an EWAS, or Environmental Wide Association Study, to look at environmental factors on a broad scale associated with disease. But at the end of the day, both of these different paradigms actually assess things one in one silo or the other. And here we'd like to ask, what about the interaction of these genetic and environmental factors? What about a combination of these factors that may confer more risk for diseases like type 2 diabetes? So why would we want to do this? Why would we want to investigate genes and environment in combination? So first of all, it's been well noted that there's this issue of the missing heritability issue. That is, for example, if I have a sister who has type 2 diabetes and I have type 2 diabetes, how much do these variants actually found in genome-wide associations actually predict whether my sibling will get the disease? And actually what we find is if we're looking at risk prediction, these variants really don't do that well. We could predict about 10 to 20% of that phenotype there. And we found about 30 loci here for type 2 diabetes. And it's growing, but these variants still don't predict the disease very well. But we also can elucidate causation if there is a true interaction. So one of my favorite examples to bring up is the PKU. So basically PKU, phenylketonuria, actually arises out of a classic gene-environment interaction here. So you have this variant that makes your hydroxylase enzyme deficient to actually metabolize phenylalanine, a common amino acid, also a part of constituents in Diet Coke and so forth, and it leads to neurological disorders. So this interplay actually allows us to elucidate these disorders better, and actually allows us a means of prevention of the disease. That is, if I stop drinking diet drinks, some of these disorders may be allayed, even though I have this genetic deficiency. So how would we like to study interaction? So I'm going to talk about this on an epidemiological scale. So I'll show a toy example, which is pretty well documented, and that's in bladder cancer. So what a gene-environment interaction means in this particular scenario is that the combination of this genetic factor, environmental factor, is different than if I had the factor, any of those factors alone. So for example, here I have bladder cancer folks in blue and non-cancer in yellow. And you see it's a combination of this environmental factor shown in green here, E+, if cigarette smoking, and in combination with this NAT2 variant here, have much more incidence of this bladder cancer than those who had either one of the factors or alone. So basically, this is a difference in outcome of this particular phenotype when individuals had both the environmental factor, that is, that if they smoked, and if they have this particular variant here. And from here, we can actually hypothesize why this phenotype might arise, or one particular pathway, is that perhaps this constituents of cigarette smoke metabolized by NAT2 lead to greater amounts of these constituents in tissue and perhaps have some mutagenic effect. And that's what these interactions as an example, may allow us to hypothesize about. So why don't we examine genes' environment simultaneously? So we actually, one of the main problems is we don't have a high throughput and low cost measurement capability for what we call the exposome, the analog to the genome. So I show here the common Affymetrix array used to assay 500,000 variants. Now we could do a million, and soon the price of these will go down to nothing, basically. But yet we still can't handle how to analyze, or we can't analyze this on the same scale for the environmental factors such as diet and nutrition and so forth, and we rely on self-report measures, which are error-prone in themselves. So if we're looking at serum-based or urine-based, tissue-based measures of environmental factors, they're going to be high cost, it's hard to multiplex, and they're not throughput. So it's very costly at the moment to get an exposome-type measure for these environmental factors. Another reason which may be of interest to us here as computational biologists is that there's an analytic complexity and large numbers of interactions that are possible. So you can just imagine, just do the exercise, it's basically G times E given a number of possible interacting pairs if we don't have any prior hypotheses. So for example, you can line up all your environmental factors here. I showed 10 sample ones, maybe diet, cigarette smoking, pesticide exposure, and so forth, and maybe 10 genetic variants. So you have the space here becomes G times E, or 100 possible interactions to test in association with your disease of interest. So you could scale this up. You have that genome array. You have 1 million variants. So this becomes 1 million times however many exposures that we consider in the exposome, G times E. And this becomes a huge burden, a huge resource burden. There's a multiple comparisons burden. And there's a large number of sample sizes that we need to uncover interactions, that it's not linear with the number of tests that we're doing. It's actually, I think it's hypothesized to be exponential through simulation by David Hunter. And also another problem, and probably a bigger, larger one, is that we choose candidate genes and environmental factors by convenience when we do these studies. So there actually haven't been very many gene and environment interactions reported, and perhaps this is because they've been chosen by convenience, and the strength of the associations are not considered. That is, the main effects of the association, the genetic factor against the disease, or the environmental factor against that particular disease. So we're not considering if their p-value is low, the strength of association, and actually has the factor been validated, and this has been pretty well documented by John Ioannidis, also a colleague of ours at Stanford. So there's need to have a data-driven way to pare down this space to test in the meantime. So how will we do that? And these factors will denote here in black that we'd like to test. So we propose this data-driven way of combining epidemiological data and toxicological data to fill in that space of where we'd like to test. So imagine you have an exposome chip with E different environmental factors here, capital E, and capital G genetic variants. What we'd like to do is, what we're proposing here is you choose a subset of those environmental factors based on their prior association to type 2 diabetes, both in the environmental and genetic side. So now you're in this subspace here. And then what we're going to do is chase down the candidate pairs in this space based on their toxicological relationship with one another. And I'll get into what toxicology is in a second. So then we'll just chase down these pairs, which is less costly to measure than the entire space of possible interactions. And so today what I'll talk about is our methodology, what types of data we're going to use to do such a study. We're going to integrate the Ann Haynes, which is an epidemiological survey, VeraMed, a new variant phenotype database that we've curated, and the CTD, a toxicological knowledge base. And I'll show you how we're paring down using the tools of EWAS, VeraMed, and actually the CTD to get to those pairs of factors that we'd like to test in epidemiological studies. So how will we integrate the epidemiological and toxicological data sets? So first, for the green stuff, for the environmental factors, we'll use this National Health and Nutrition Examination Survey, and we'll crank a EWAS on type 2 diabetes to get the environmental factor candidates. Then we'll use VeraMed to curate the literature of all the genetic variants that are associated with type 2 diabetes, and we'll get the red factors there, the genetic variants associated with type 2 diabetes. And there we've got a putative space of interactions that we'd like to test. Then we'll use the comparative toxicogenomics database, which links genetic factors and their products to environmental factors to peruse this space of possible genetic and environmental factors to test in further studies. So first I'll talk a little bit about the Ann Haynes. If you don't know, maybe you're part of this study. It's actually a study conducted by the CDC, which is a fairly comprehensive one about the nations, the U.S. nations' health and nutrition. So basically, it's a publicly available, the data is publicly available, conducted every 2 years, and it's been around since the 60s. It's been instrumental in providing standards for growth and development for children, and also the trending of how the health of the nation has trended through time. So what they do is they take these trucks here, which are manned by health professionals, and they go to 15 different places around the United States, and they'll sample people in different counties, and they'll actually do a comprehensive phenotypic and environmental assay on these individuals. So basically, so some of these phenotypes that they'll collect are quite rich. You'll get maybe fasting blood glucose, urinary creatinine, and they'll ask them a pretty comprehensive health questionnaire about what they feel, what their self-reported health status may be. You'll get a rich set of demographics information, such as race, socioeconomic status, sex, and age. And of interest to us, you'll get about a large amount of environmental exposures assayed in human tissue, that is in serum and urine and even hair. So in lieu of that exposome chip that it's so elusive right now, we'll use the Ann Haynes to actually assess these environmental factors against our disease of interest, such as type 2 diabetes. And so different factors that are measured in Haynes, you got markers of smoking, of cigarette smoke. You got direct markers of nutrients and vitamins, such as vitamin A, B, C, and carotenoids. You have markers for allergens, such as against cat hair, dog hair, grass, and so forth. You got chemicals and consumables, such as bisphenol A and phthalates. And actually, just today, the bisphenol A was banned by the FDA to be put into baby products, so already this slide is getting old. Industrial chemicals, such as polychlorinated biphenyls and hydrocarbons as a byproduct of combustion. Pesticides and herbicides that are applied to the foods that we eat, and even infectious agents such as HIV, hepatitis, and so forth. And so their prevalent exposures measured in individuals in the US, you can think of them as common variants, if you will. They're probably a mixture of these things in all of us sitting here in this room today. So now what we'll do is actually talk a little bit about VeraMed and how we select variants that actually are connected with phenotypes. So the literature of variant disease association studies, unlike environmental studies, is growing. It's vast. And there's actually over 200 of these studies done for type 2 diabetes alone if you just count the GWAS literature. So here's some samples here, just beginning in Nature with this large WTCC, the Wellcome Trust Case Control Consortium, 2007. Shortly thereafter, you had this paper in Nature by Slodek and colleagues looking at type 2 diabetes, a validation study in Science, and so forth. And the list goes on. And what we've done here is with VeraMed, we've actually comprehensively curated all of these findings in our lab here. So what VeraMed consists of is about 7,000 papers identified via mesh, and the terms that we're looking for is genetic association or GWAS and disease and so forth. We have over 100,000 variants and mapping to about a third of the genome. We have looking at many phenotypes like obesity, height, type 2 diabetes, and so forth. And just to note, it's actually a superset of NHGRI's quite good GWAS catalog. So you have more variants in there than the GWAS catalog that a lot of folks rely on. So the data would look like this. So for a particular paper, you might be looking at a phenotype that was measured in that particular investigation. You'll get a RSID or a dB-SNP. You'll get the risk allele, the effect size of that particular finding there, and a cohort of interest. And so this is important for doing these types of interaction candidate analysis where we need to know what cohort these were measured in. And so we'll have different phenotypes measured here, like body mass index and so forth. And at the end of the day, you have about 1 million rows of these variant phenotype associations here. So next, I'll tell you a little bit about toxicology and CTD and these gene-environment relationships archived in this particular knowledge base here. So just in brief, toxicology, as you may already know, is the biological response or the physical response of environmental or other physical factors and genetic factors conducted in model systems. So here I'll show you an example from Som et al., and they're looking at bisphenol A, that same factor I just told you was banned, and they're looking at this particular phenotype, adipogenesis in rat, and it was published in this journal that is very known in environmental health. So what they're looking at is basically high doses of bisphenol A applied to rats, and then they're going to observe the molecular relationship between that given exposure and the phenotype that they're observing there. And so what you could code that is bisphenol A leads to higher expression of this particular gene, LPL mRNA. So toxicogenomics actually examines this response on a genome-wide level. So actually, you can abstract this relationship. So bisphenol A leads to higher expression of LPL mRNA, and you can actually curate all of these findings in a computable format. And this is exactly what the CTD is. So in effect, it consists of 6,000 environmental or physical factors in their relations with 21,000 genes of interest, and about 200,000 of these relationships, such as leads to higher expression of or changes the expression of gene products such as mRNA and proteins and so forth, on over 300 organisms such as mice and human cell lines and so forth. So now you have a connection between an environmental factor and a gene. So now you can see which factors that you found in your GWAS investigation or your EWAS investigation to see which ones have a biological relationship. So in summary, the Ann Haynes is what we'll use to crank environmental factors associated with disease. It has about 250 exposures, over 200 phenotypes, and over 5 race groups. And VeraMed which we'll use to grab genetic associations for type 2 diabetes for folks of European ancestry. And we'll use the toxicogenomics database to connect these environmental factors to genetic factors with real toxicological curated relationships between those genetic and environmental factors. And so you can ask the simple question, actually, before you begin that investigation to predict candidate gene-environment interactions, is actually how many of these genetic factors that we've associated with complex disease have some biological interaction in the CTD. So we've actually, that's a simple join there, and you can actually see for all the diseases that we have in VeraMed, for the complex diseases with a large number of genes implicated in GWAS, a majority of them, shown here, a percentage of those genes that are involved in a biological interaction, are quite large. So for example, the metabolic trait here has about 90% of its genes implicated in GWAS, I think of greater than 150 genes implicated in GWAS, that actually have some documented biological relationship in the CTD. And one thing to note here, if you look at type 2 diabetes, about greater than 30 genes have been implicated in GWAS, and 100% of those genes actually have a biological or toxicological relationship documented in the CTD. So based on this, we thought it would be a great knowledge base to produce candidate gene-environment interactions for type 2 diabetes. So I'll just tell you a little bit how we conduct EWAS to pare down the environmental factors that we might look at. So what we do is we use that Ann Haynes database I told you about earlier, and we identify cases and controls. Here we're going to use the American Diabetes Association threshold for type 2 diabetes of 126 milligrams per deciliter. And we're going to focus our cohort to non-Hispanic whites, since most of the variants that we've found in VeraMed actually have been measured on folks of Caucasian or European ancestry. And then we'll actually take all those environmental factors I told you about, markers of cigarette smoking, of nutrients, and so forth, and associate that with type 2 diabetes, whether the individual had it or not, using a logistic regression. And we'll adjust out for things that could bias our results, such as age, sex, socioeconomic status, and so forth. We'll collect the size of the association there, the beta factor or the trend there. And we'll also compute a false discovery rate. As you know from Stats 101, we're conducting multiple hypotheses here, so we'll compute the expected rate of false positives here using the false discovery rate. And we do that empirically by shuffling the cases and controls. So at the end of the day, this is our EWAS Manhattan plot. So if you're used to looking at GWAS Manhattan plots, this is one we use for the other side of the fence, for the environment. So our chromosomes are now our categories of environmental factors such as nutrients, cigarette smoking, phytoestrogen, hydrocarbon exposure, heavy metal exposure, and so forth. And the y-axis here is your strength of association here. So what we're looking at is basically the association to fasting blood glucose adjusted by BMI, SES, age, and sex. And the odds ratios I'll show you in a second are for a standard deviation change in exposure, so we can compare them across the board. So then here's our line of multiple hypothesis correction of FDR of less than 10%. You could pick various thresholds, which I'll show you in a second. And at the end of the day, you get factors such as heptachloroepoxide, a pesticide banned in the 70s associated with type 2 diabetes that we were able to validate. PCB 187 has a pretty good observational epidemiological evidence with association to type 2 diabetes. Odds ratio about 2, so roughly 2 times the chance of being type 2 diabetic in this cohort. Beryllium, a heavy metal that was also omnipresent in the environment associated with type 2 diabetes, and a whole host of nutrients, including gamma-tocopherol, which is a type of vitamin E, cis-beta-carotene, which is a carotenoid, vitamin D, and also other forms of beta-carotene, such as trans and total beta-carotene associated with the disease. So interestingly, gamma-tocopherol has an odds ratio above 1, which is associated with having the disease for greater amounts of gamma-tocopherol, and beta-carotenes were protective for the disease, as was vitamin D. And so now we actually will collect variants associated with type 2 diabetes to grab the genetic factors that are of interest. So when we do that, we selected those that were for type 2 diabetes, and we found that for nominal associations, for basically 0.05 without multiple hypothesis correction, you have about 690 variant, almost 700 variants in there, mapping to about 360 genes. And if you're strict about it and you look at only those variants found in GWAS and so forth, we have about 75 variants in there, mapping to 35 genes. Then you can just run the exercise of seeing which one of these factors have a gene-environment relationship in the toxicological literature in a high-throughput fashion. So that's what I'll talk about next here. And so you could produce a plot like this. And so basically on the x-axis here, I'm showing you the strength of association for the genetic factors, the red factors I showed earlier. And the y-axis shows here the number of hypothesized candidate gene-environment interactions you could peruse through. And the different colors I'll show you in a moment correspond to different EWAS FDR levels. So if you're extremely non-stringent about your thresholds of what you consider to be strong main effects, suppose you say p-value 0.05 for genetic effects and FDR of 100% for environmental effects, we predict in the CTD there are about 5,000 different types of biological gene-environmental interactions you could examine in the context of type 2 diabetes. And then you can actually explore this space based on your threshold of what you'd like to believe is true. So for example, if you only believe that GWAS results are true, and so you could look here in this basically in this regime over here, and you only believe that FDR of 10% is good for validated factors, you can look here and see that roughly about 6 factors are predicted in the CTD of between interactions, or between genetic and environmental factors found in EWAS and GWAS studies respectively. So then you could actually use these factors in prospective studies to actually look at the interaction between these factors in context of type 2 diabetes. So I show you some of these factors that we predict that we'd like to chase down in further studies. Chosen by that threshold, I show EWAS of FDR of 10% and a GWAS significance level for genetic factors. So the first one includes gamma-tocopherol. It had an odds ratio about 1.4 in its main effects in our EWAS. And if you look at the CTD, it actually shows increased expression for PPR gamma, which was also implicated in GWAS, in VeraMed. Now, also the carotenoids seem to be have evidence of actually interacting with PPR gamma. So here in this Kawada study, it showed that the application of cis-beta-carotene to rats actually decreased expression of PPR gamma. And it's interesting to note here that the relationship between these odds ratios, below 1 and above 1, seem to be concordant with what we see in the literature in 2 different studies here, that they decrease expression and increase expression of this particular mRNA genetic product here. Vitamin D here seems to be affecting expression in this particular toxicological study. And PCB 187 seems to be messing with PRC1. Actually, was found in also found in GWAS with an odds ratio of 1.1, and this PCB factor was found at an odds ratio of about 2, and it increases expression. So, at the end of the day, what this list provides us is a tractable a list of factors that we could test in combination in association with type 2 diabetes that we have strong or some biological prior of their physical interaction in human systems. So we actually, I won't get into the details of this, but we actually tested the significance of getting this set through some randomization tests, and we estimate getting some of these about 5 of these factors in this particular list was about a p-value of 0.001 here. So at the end of the day, to conclude, I've showed you how to traverse this space of genetic and environmental factors, integrating environmental and genetic epidemiological data with findings from toxicology to produce these candidate genetic and environmental pairs to test in further studies. So we've created these genetic gene and bioenvironment interactions in type 2 diabetes to test, and these include gamma-tocopherol and beta-carotene with PPAR gamma, and also vitamin D, and also PCBs and PRC1, with some reasonable biological relationships following them. So, but what we did was actually we chose thresholds to choose this G and E based on their strength of association in their respective studies. And so what might happen is we may miss potential interactions. But what I posit is that actually these are prioritized by using these thresholds by their relevance, especially the environmental factors and their public health relevance. So these are strong associations because they exist throughout these populations in Ann Haynes. And so this way we actually are prioritizing by their public health relevance. And also this provides a less analytical and actual cost of measuring these candidate factors. Hopefully someday we may have an exposome chip where we can measure all of these things in tandem, nevertheless. And there's actually a possible bias by using these data sources. For example, I'm not claiming that we're measuring the entire exposome. The EWAS is a limited number of exposures that we're measuring in association with type 2 diabetes. And of course, it's a cross-sectional data set. There could be some confounding bias and so forth. And in the CTD, there's some investigator or study bias in which they are actually going after some toxicological relationships that are notorious in the literature, such as bisphenol A or cytochrome P450 genes. So there's these file drawer and also these cross-sectional observational studies that bias that we may have to overcome. But nonetheless, we feel like these are good candidates for further study. I'd like to thank my co-authors here at Stanford, Rong Chen and Atul Butte, the ISMB for the travel fellowship, and of course the Farmer Foundation for the funding. And thank you for braving through the last session here. Thanks. I'll take your questions.
Okay. Great talk. I have one question. Can you extend directly your approach to beyond binary interactions, for example, 2 genetic ones and 1 environmental, or do you believe that the current cohorts you're mining are prohibitively small to do that?
So you mean 3-way interactions and so forth?
For example, yes.
Yeah, I actually haven't thought of that, but I think I get a little apprehensive of testing those, especially in epidemiological cohorts where the sample sizes the power needed is going to be large, very large. Great, thanks for your attention.